Well, good morning, everyone, and welcome to this week's edition of the 2023 Speaker Series. This week, we welcome Dr. Carolyn Strömberg, and I would just like to extend a thanks to the Royal Truro Museum Cooperating Society, who generously fund the Speaker Series. So Dr. Carolyn Strömberg is a paleobotanist and a paleoecologist and is currently the Estella B. Leopold Professor in the Department of Biology at the University of Washington and curator of paleobotany in the Burke Museum of Natural History and Culture in Seattle. Dr. Strömberg's main interest is elucidating the evolution of grasses and the assembly of grassland ecosystems during the last 100 million years. But she is also broadly curious about how plants and plant-animal interactions have responded to environmental change in deep time, and what that can teach us about ecosystem function today. Research in her lab at the University of Washington involves fossils and extant plants, phylogeny, morphometrics, and modern analog work. So everybody, I present to you Dr. Carolyn Strömberg.
Thank you so much. I'm very excited to be here today, speaking to you. And I'm going to talk about some work that we're doing to elucidate, like Chris was saying, one of the largest, is maybe not the largest, ecosystem change since the extinction of the non-avian dinosaurs, about 66 million years ago. So, as Chris said, I am a paleobotanist, which means that I study old plants, so paleo being old and botany being the study of plants. And I've studied both fossils that you can see with the naked eye, so megafossils like this lovely leaf here on the right-hand side, and also microfossils, so fossils that we can only see using a microscope, such as this silica plant fossil, and I'll talk more about that later, what that is, on the left-hand side. And I'm mainly going to be focusing on work using these microfossils. So, as again was said in my lab, we're broadly interested in the role that plants have played in the evolution of terrestrial ecosystems through geologic time, so through millions of years, and how we arrived at these all these diversity of ecosystems that we see around us today, how that was driven by changes in the abiotic environment, so climate, the atmosphere, how continents have moved, and also how that influences in turn other organisms on Earth. We have focused a lot on the evolution of a particular set of ecosystems in the so-called grassland biome. So this is savannas, temperate grasslands, woodlands, and so on. So when I talk about grassy environments, I mean all of those things, unless I specify something more detailed. And why are we so interested in grasses and grasslands? Well, it is because grasses are unique and important. They are the fourth most speciose plant group on Earth today, or plant family, I should say, with about 12,000 species. And just to put that in context, living mammals today are about 6,500 species. And I don't know if this has been updated, but the number of non-avian dinosaurs recognized in the fossil record is about 2,000. So a lot more of grasses than any of these charismatic organisms. They also have very diverse growth forms. So everything from a minute herb here, Mybora on the left, to giant tree-like bamboos that can be up to 100 feet sometimes. So very large. Grasses also live in very diverse climates, so everywhere from the tropical rainforest to hot and cold deserts. And in fact, they're a grass that's one of the few vascular plants that are now permanently living on Antarctica, which of course is a sign that climate warming is really happening. So not a very fun thing, but it does show how versatile grasses are in terms of their climate space. Grasslands are also very unique and important. So grassy biomes are incredibly important, making up over 40% of Earth's land surface. And you can see grassy biomes here marked in different colors on this map. And they provide some really important things for us and other organisms. So over 1 billion human beings live in grasslands and many, many millions of other animals as well. They have an influence on climate because they absorb sunlight in a different way compared to forests or ice. So they influence the distribution of heat that way on Earth. And they also have a different relationship with water compared to forests and deserts. So they influence how water is distributed on Earth. And also they play a big role in geochemical cycling, so how carbon is moving through the environment. So how it gets fixed, how it gets stored. And this in turn has a feedback sort of influence on climate as well, because CO2, where the source of carbon is, of course, a greenhouse gas. So important for Earth in general, but also important for us humans specifically. We have grasses were one of the earliest crops to be domesticated some 12,000 years ago on different continents, and is still make up the largest part of what we eat today, including corn syrup here in North America, and also important building material in the form of bamboo, most of what our domesticated animals eat. And here's an example of a grass as a biofuel, switchgrass. Also, many of our most important weeds are grasses, so it's not all good, but it does mean that grasses play a really important part in our lives as humans. And so given their importance today, paleontologists and evolutionary biologists have long been interested in when and how grassland ecosystems evolved. They're so important today. We know they were not around when the dinosaurs ruled the earth. So it happened very recently. So how and when did it happen, and what were the driving factors? And this is not just a sort of a curiosity for knowing Earth's history, but it actually plays a fundamental part in understanding how grassland ecosystems function today and how they might change in the future. So to give an example of that, here's the distribution of savannas as black O's here versus forests as orange X's in climate space across continents. So on the x-axis, we have mean annual rainfall on these different continents. On the y-axis, we have the length of the dry season. And you can see that across continents, we see similarities in the distribution of these different vegetation types in climate space, but also some differences. So generally speaking, forests tend to be at higher rainfall, but there are also some big differences here. And those differences have been suggested, or researchers have suggested that relates to the kind of bedrock that's there. So the types of rocks that form the landscape where these different vegetation types are growing, the kinds of trees that evolved in these different continents, the kinds of animals that occupy these different continents. So in other words, it depends on the history of those biomes on these different continents. History matters. So history matters for how these vegetation types are responding today in climate space, and likely also how they will respond in the future. So really important to understand at this point in time when we are seeing major climate changes happening because of humans. Okay, so we're interested in reconstructing these past grasslands. What kind of evidence can we use? So people in the past have looked at, for example, grass pollen, putative grass leaves, and grass fruits in the fossil record. And it turns out that those kinds of fossils are rather rare until the late Miocene. And the ones we have are, for the most part, ambiguous. So leaves are very hard to interpret without preservation of anatomy, so cells. And so for the most part, we can just say, it looks kind of like a grass leaf, but it could also be a sedge or a cattail or some other monocot. And then grass pollen are very similar to pollen of their closest relatives. So we can't know for sure if we're looking at grasses all the time. So instead, people, when they want to reconstruct grasslands, have looked at indirect lines of evidence, in particular at fossil faunas. So there are animals today, like these zebras, that we know live and feed in grasslands, and they have particular functional traits, so shapes and morphologies that relate to their function. And in particular, people have looked at the evolution of cheek teeth, how their crown height has changed has evolved within large tooth mammals or ungulates. So here we have a human tooth, which has a very short crown. That's called brachydont. And here we have a horse tooth that has a very tall crown. That's called hypsodont. And we have these hypsodont cheek teeth shown in the skull of a young horse here on the right. And so what we notice today when we look at ecosystems, as I alluded to, we tend to see herbivores with these hypsodont teeth in grasslands and other open habitats. And so the assumption has always been that it is because it's a way to deal with feeding on very abrasive food materials, such as silica-rich grasses, because grasses have a lot of silica in their tissues, as we'll talk about, and also because when they're feeding in low to the ground or in open habitats with a lot of bare soil, they tend to get a lot of dust and soil in their mouth, and that also wears their teeth. And so the significance of this is that these the silica in the grasses and this grit wears down the teeth. And that means that the more crown you have, the longer you can live, the longer is the functional life of the tooth. So it is an advantage in those grasslands. And the assumption, therefore, has been made that evolving these tall tooth crowns was an adaptation to feeding in grasslands, either on grasses or grit. And using this line of evidence, paleontologists have looked at when in time did hypsodonty show up on different continents, and use that as a mark, as a signal that at that time is when we must have started to see grasslands spreading, because and that those animals evolved hypsodont teeth in response to that. In my lab, we wanted to test this by looking at plant fossils, because here so far we have no plant fossil evidence that can be easily compared to the animal evidence for the animal indirect evidence for grasslands. So what we're using is plant silica fossils, microscopic or fightlets. And so what are fightlets? They are tiny microscopic silica bodies that form inside and around cells in living tissue. This is the outermost layer of a grass, the so-called grass epidermis, a leaf. And inside some of these cells, we see these silica bodies forming. And then when that leaf is dying or decaying, those silica bodies are left in the soil or the sediment and can stay there for a long time, because it's chemically much more it is not as reactive as organic material, so it can stay there for a long time. It's also very hard and durable. So we can use then these fightlets that are staying in the sediment or the soil to say something about what plants were growing at that particular site. And so if we compare fightlets to other types of paleobotanical data, you can't get very specific information about, for example, the types of trees that were in a particular place, but we can say something about the broad vegetation type. So whether we're looking at a savanna or a forest. And this is, of course, what we want to know when we're trying to reconstruct how grasses are becoming more abundant in the environment. So they're very good for that. Also within the grasses, so this is a picture here of a grass family tree, or a phylogeny as it's called. So showing how different grasses are related. We also know that there is variation in the shape among fightlets across the grasses. And that means that we can say something, we can look at fightlets in the fossil record, grass fightlets, and say which kind of grass it came from, which subgroup of grasses it came from. And this is important because, as I'll show you in a second here, different grass subgroups have different ecologies. So they can say something about ancient grass communities, what kind of grass community, what was the ecology of that grass community. Another really cool thing about fightlets is that they can be preserved in the same sediments as fossil faunas. And this is unique, I would say, to fightlets, because in many of the sites where you find faunas, you don't find other types of fossil plants, such as leaves or pollen. they're often been oxidized away. So this is good because we can get a direct paleoecological link between floras and faunas using fightlets for that reason. And we can capture some part of the environment that we don't see when we look at other types of fossils, potentially. Okay, so before we dive into the research, let's just familiarize, so we're all on the same page when it comes to grasses. What are they? So, or what can we say about different kinds of grasses? So we know that the initial radiation occurred either of grasses. So we look down here at the bottom, the most early diverging grasses. We know that those today live in relatively close habitats, so either in the forest understory or in forest margins. And so we know that they probably sort of initially lived in closed habitats. Also, there are some groups like bamboos, for example, that still live in closed habitats. And then there were two groups that independently evolved the ability to thrive in open, drier habitats. One was the cool temperate poids here, the poidae, and one was the tropical packmads. And within this packmads is where we find some of our most common savanna grasses, for example, tropical grassland grasses. Okay, so using fightlets, I'm going to talk about 3 different questions that we've been able to address in the lab. One is, when did the grass family first originate and diversify? When did open habitat grasses diversify and spread? And how did the evolution of grasslands relate to the evolution of grass-eating herbivores, those hypsilon herbivores? Okay, so first I'm going to talk about some work looking at this first question. This is collaborative work with colleagues from India and elsewhere. And it's looking at fightlets from late Cretaceous sediments in central India. So what we did was we went to some sites in central India that had been dated to just before the extinction of the non-avian dinosaurs, about 66 to 68 million years, perhaps, something like that. In those sediments, we have certain horizons that weather out in the form of agricultural fields, and where you can find dinosaur bones, such as this one, turtle bones, all kinds of bone scraps, but also a bunch of fossilized poop, so-called coprolites. And here are some examples. Some are 5 to 10 centimeters in diameter, sort of blobby things, but there are all kinds of other kinds as well. These larger ones have been most reasonably linked to dinosaurs, herbivorous dinosaurs. But these coprolites, the whole assemblage of coprolites, have been studied since the late 1930s by primarily Indian researchers. And what they found inside them is that they contain abundant organic material, including from bacteria, algae, and fungi. But they've also found a lot of tissues and also pollen or spores from conifers, cycads, ferns, and horsetails. So indicating that these dinosaurs were feeding on a broad range of plants. Okay. We took these coprolites into the lab and macerated them, basically broke them down with chemicals, and then we saw what we found. And what we found was rare remains from insects. So this is a jaw element here of an insect, plus some fightlets, both from palms, dicots, and ferns, but also from grasses. And this is the most important and exciting thing. Here is showing some images of these lovely fightlets. So just to orient you here are some typical grass fightlets. We call these short cell silica fightlets. And then here are some silicified, basically epidermal cells around that. And also in some cases, some cuticle. And cuticle is the waxy outer layer of a plant. But we found a whole range of these different types of grass fightlets. We've continued to work in India, looking not just at coprolites, but also at different sediments from the same time period in central India. And there we continue to find fightlets. And and whole silicified pieces of epidermis, where we could get a lot of sort of, characters, a lot of information from those. And so what we did was we looked at a whole bunch of modern grass fightlets and epidermal features, and then we compare that to our fossil ones. And then we use that in combination with DNA molecules to try to figure out where in the grass phylogeny, so where in the family tree of grasses, these fossils fit. So we did a so-called phylogenetic analysis. And we found, so this is the family tree that we came up with, we found that these fossils from India fit in the Oryzae, which is the rice tribe within the grasses. And so that then allowed us to say that this tribe must have arisen by 66 million years ago, because that's the age of the fossil. And that then allowed us to date the entire tree, the entire family of grasses. And what we found was that grasses evolved much earlier than previously thought in the early Cretaceous, maybe some 125 million years ago. And that was up to 45 million years earlier than previously thought. So that was exciting. It also has led us to think a little bit about where grasses evolved. So our fossils are from India, but there's also some grass pollen found in northern South America and in Africa. And so that led people for a long time to say that grasses probably evolved in these southern continents, so-called Gondwana land. And that they had done so before the late Cretaceous, because at that point they were already widespread across these continents. Now, just a few years ago, there's some fossil fightlets described from China, and those fossil grass fightlets were 100 million years old, so significantly older than the previously studied grass fossils, both the fightlets and the pollen. And they were actually found in the teeth of a hadrosaur, so definitely associated with dinosaurs. So this then calls into question whether grasses actually evolved on the southern continent. So now we really have to intensify the study and try to find fossil grass fightlets and other grass fossils on different continents to try to figure it out. In the meantime, one of my grad students, Elena Stiles Rosselli, is looking at early grass fightlets from southern South America, from Patagonia, and tried to figure out what grasses were around there and what habitats did they live. And based on that, what can we say about how grasses diversified and spread across continents during the early days. The other thing that we could conclude based on our study is that certain dinosaurs fed partly on grass, but that grasses were only a small component of a very varied diet. And the herbivorous dinosaurs that were around at that time were titanosaurs, rather small sauropod dinosaurs, but potentially they were feeding on a very broad set of vegetation. Another cool thing is that in India and other southern continents, there's this marmot-sized mammal called Gondwanatherian mammal, and they have very hypsodont teeth. And previously, the idea that they may have fed on grasses had been discounted, but now, again, that is a hypothesis that we have to consider. Okay, so now let's switch gears to looking at when open habitat grasses diversified and spread, and how did that relate to the evolution of grass-eating herbivores. So the spread of grasslands has long been thought of as a response to changes in climate during the last 66 million years. So to orient you here, we have time moving forward on the y-axis as we go up. And then on the x-axis, we have global climate reconstructed using oxygen isotopes from shells of marine microorganisms, so-called foraminifera. And basically what we see here is that we have very high temperatures in the early Eocene here, around 50 million years ago, and then we see a decline with some bumps in it. And so we have increasingly cool climates on Earth. Also on mid to high latitudes, we see increasingly dry climates for various reasons. And so the idea was always that grasses diversified and became abundant as this was happening, and that happened in coevolution with herbivores that grew larger, grew hypsilon teeth, and diversified and became abundant. So there was this idea of the coevolution between grasses and herbivores during this time. So this is something that we've wanted to work on and address. And this is just showing some of my many collaborators on these projects trying to reconstruct the spread of grasslands and the evolution of hypsilonte. I'll be talking a little bit about these three areas, North America, Eastern Mediterranean, and Patagonia as we go here. First, let's talk about North America. This is where classically the evolution of horses happened after horses, or the earliest horses immigrated from Asia around 55 million years ago. North America has just a tremendous fossil record of this evolution of horses, and in part because of the many collecting trips that O.C. Marsh and others took during that time. And I don't want to get into the horrible atrocities that happened against Native Americans as a result of that. But one positive thing is that we know a lot about horse evolution as a result. And what we know here is that we see the evolution from these tiny early eohippus that were cat-sized and have very low crown teeth, all the way to our large, long-legged, and very hypsilon equus today, the only living genus. And we see this diversification. And this was then thought of as a response to the spread of grasslands. And this is shown here in this image from Simpson's book on horses. And based on it, researchers pinpointed that sometime between 18 and 15 million years ago is likely when grassland spread, because that's when we start seeing the evolution of hypsilonte really taking off within horses. This is something that we wanted to test. So I analyzed phytlet assemblages extracted from Eocene through Miocene into Pliocene sediments, so 40 to 5 million year old sediments from the US interior. And not just me, but me and my students and colleagues as well. This is showing one example of the kinds of areas. So we've tried to focus on areas where animal fossils have been found as well, and that were well dated. So before we dive into the data, I just want to talk a little bit about how we analyze vegetation based on phytlet assemblages. So this is showing, this pie chart here is showing a fossil phytlet assemblage. And what we do is basically to reconstruct vegetation structure or habitat openness, is that we compare the amount and types of open habitat grass phytlets compared to phytlets indicative of plants that tend to grow in association with or in forests. Okay, so that's the simple way of saying it. And we also look inside the grass communities to look at closed habitat grasses, and those also help us indicate that we're looking at a forest habitat. So the very coarse rule of thumb is that in grasslands we tend to see well over 50% open habitat grass phytlets, and this will become important later on. This is just showing some of the pretty phytlets that we have. So this is phytlets indicative of palms, a range of phytlets indicative of woody and herbaceous dicots, so angiosperms, conifers, and gingers. And then we have our bamboos and other types of closed habitats, so forest grasses, and that's compared to then the open habitat grass phytlets. Okay, so let's dive straight into looking at North American grassland evolution using phytlets. Here we have, again, time on the y-axis moving forward as we go up. And then we have data from Nebraska, Wyoming, and from Colorado. So the central Great Plains, if you will. What we see here is some clear patterns. First, I just want to point out that there's some uncertainty in timing and some lack of data from some of these time periods and regions. But basically, what we think we're seeing is that the Eocene, Oligocene was dominated by forests. So these pie charts or these assemblages are dominated by forest indicators. Grass communities are by and large dominated by bamboo fightlets, or what we interpreted as bamboo fightlets. And then we still, during this time, we still see minor abundances, and in some cases more higher abundances of typical looking open habitat grass fightlets. This is a poidy fightlet, for example. And this is happening in the late Eocene. So this indicates to us that even though these are primarily forest habitats, we have open habitat grasses present either in the understory or in gaps or at the margins of forests. And then we see sometime during the late Oligocene or early Miocene, depending on the dating of some of these assemblages, we see an expansion of these open habitat grasses into open grass-dominated habitats, maybe woodlands, maybe savannas, most likely some kind of mosaic of more forested patches and more open patches. And this is happening, like I said, in the late Oligocene or early Miocene, maybe 25 to 22 million years ago. And importantly, these earliest grasslands were dominated by cool temperate poid grasses. These are grasses with the ancestral photosynthetic pathway C3, but we're not going to talk very much about that, so you can ignore that. We also have looked, so now I removed Colorado. We've also looked at Montana, Idaho. So this is a little bit further north. And we see there a similar pattern, actually, where we see forests, more closed habitats in the Eocene and Oligocene. The difference here is that we have a different set of grasses. We're not entirely sure if they are closed habitat or open habitat, but primarily we see closed habitats here. Maybe with some more local, more grassy habitats. And then sometime in the late Oligocene or early Miocene, we see the expansion of typical open habitat grasses forming some kind of open grass-dominated habitats, also in the northern Rockies, so the Montana and Idaho. And so this indicates to us that this shift, it's actually seems to be fairly synchronous between these regions, indicating that some large-scale change must have happened to allow these open habitat grasses to expand, and we'll get back to that. So what are these data showing? They're showing that the taxonomic diversification of grasses, of these open habitat grasses, is happening down here when we see our earliest open habitat grasses, but it's not until some 20 million years later, actually here in North America, it's more like 11 million years later, at least, that we see the expansion, the ecological expansion of open habitat grasses. So when they become ecologically dominant. And this indicates then that there were different drivers between the taxonomic diversification and the ecological expansion. And what were some of these drivers then? We have collaborated with some geochemists, Tyler Kukla, who used to be at Stanford, but is now actually doing a postdoc at University of Washington, and some other folks. And what they did was they looked at the isotopes, the oxygen isotopes of clays and carbonates. So these are different minerals in the sediment or the soil. And this allows us to say something about rainfall of seasonality. So they looked across this Western US here, including the regions that I talked about, and they took samples across those regions and compared it to the fightlets data down here. And to make a long story short, they found that climates in the continental interior became more arid in the winter during the late Oligocene to early Miocene. So we see an increase in winter aridity during that time. So this seems to coincide with this opening up of landscape and the spread of open grass-dominated habitats. So we are hypothesizing that change is what drove that. And we are further hypothesizing that it may have been the rise of the Cascades in the West here that helped create this gradient of aridity across the Western US. Okay, what about the evolution of grasslands in relation to the evolution of Hypsidanti in North America? What we see is that Hypsidanti evolved at least 4 million years after the spread of grasslands. And so even though we think that Hypsidanti could still be an adaptation to these new open habitats, there doesn't seem to be a tight coevolution between grasses and herbivores. The take-home message, though, is that these herbivores evolved in the context of open grass-dominated habitats. If we look at the Eastern Mediterranean, we see a similar pattern of a long delay, several million years, between the spread of grasslands and the evolution of hypsodonty. But again, the idea here is that hypsodont herbivores evolved within grasslands or grassy habitats. Now, we were interested in looking at South America as well, because in South America, we have an endemic group of hoofed mammals that are now entirely extinct, so-called southern ungulates or meridi ungulates, and they evolved increasing hypsodonty and something called hypsilodonty, which is basically ever-growing teeth, so an extreme version of hypsodonty, during the Cenozoic. So if we look here through time, we see that hypsodont and hypsilodont cheek teeth increase through time. And this is looking within the Noda ungulate group, which is a subgroup of these southern ungulates. So we were interested in looking at that. And just to say that we see this change happening very early on, the evolution of hypsodonty, so 20 million years earlier than on other continents. And so the idea had been that the earliest grasslands on Earth actually spread in South America in the middle to late Eocene, around 40 million years ago. So this is something that we wanted to test. And this is the team that set out to test that. And the most important person here is my now former grad student, Reagan Dunn, who is now at La Brea Tar Pits as a curator of paleobotany there, but also a whole group of vertebrate paleontologists and geochemists here. So we were interested in looking at whether open grass-dominated vegetation occurred prior to or coincident with the first hypsodont herbivores in southern South America. And the test then, of course, is to reconstruct vegetation based on fightlets and compare it to the faunal record. So to do that, we went to a place called Gran Barranca in Patagonia, Argentina, which is this very long section of rocks that spans 42 to 18.5 million years, so Middle Eocene through the early Miocene, and it has 80 faunal levels, so a lot of faunal evolution is recorded in this section, and the sediments here are modified volcanic ash, and this will become important later on. So we sampled extensively for fightlets and analyzed a whole bunch of samples from there, and the preservation is primarily quite good. So that's cool. And we used a modern reference collection to classify them, of course, as we do. And just to remind you of these different things, because now we're going to look at a fightlets assemblage as a bar graph. We have palms, we have gingers, spiral gingers and relatives. These are tropical herbs that tend to be associated with forests. And then we have other forest indicators, woody and herbaceous dicots, and so on. We have closed habitat grasses, and we have our open habitat grasses and dapoids and packmats. Okay, so then let's look at the data. So here we have time moving forward on the y-axis as we go up, and we have our fightlets assemblages as bar charts here. And what we see, a striking pattern here, is that grass fightlets are less than 25% of any given assemblage, even though we see grasses present from about 43 million years ago. And by 40 million years, we have representatives of modern clades such as bamboos and also open habitat grasses, poets, and packmats. But these grasses are quite rare, relatively speaking, and we have primarily a lot of palms and a lot of woody and herbaceous dicots. So we interpreted this initially as some kind of forest. And this vegetation type seems to persist through up here, through the Oligocene. And then in the Miocene, we start seeing more abundant open habitat grasses, up to 25%, but still primarily dominated by other plants. So we still interpreted this as forested, but with maybe some open grassy areas. We looked at Gran Barranca. We've also looked a little bit higher up in the section, so at younger sediments. And we found a similar thing here, that grasses are mainly rare on the landscape prior to about 11 million years. We haven't analyzed anything younger than that, so that's what we know. And so we concluded then that grasses were not abundant in southern South America prior to about 11 million years ago, so the early late Miocene. And this is counter to previous fightlets work that had been done in that region, but it is consistent with other paleobotanical data from the same region, in particular fossil pollen, which indicate that it's not until after about 10 million years that we see steppe taxa, so grasses and other types of pollen, dominate over the forest taxa. Okay, so what about Hypsodonty evolution in Patagonia? So we can conclude that Hypsodonty in South American ungulates did not evolve as a response to the spread of grasslands, because there were no grasslands when Hypsodonty came around. But what do we know about openness? So I told you that we interpreted this primarily as forests. And this is because in traditional phytolith analysis, we use the presence and abundance of open habitat grasses as an indicator for the openness, basically. But what about if you're looking at a habitat that is very open, but where grasses are not dominant, such as this shrubland here? It's not clear that traditional phytolith analysis can actually detect that in the fossil record. So with Reagan, we set out to come up with an alternative way of inferring openness in the fossil record using phytoliths. And Reagan came up with this idea that we could use that there's variation in plant traits with light. So, for example, if you look at leaves growing in the sun have a different size. They're smaller and they are less lobed than leaves growing in the shade, because that is how they optimize their surface area and so on for the light. And similarly, when we look at the epidermis of plants growing in full light versus shade, we see differences in the shape and size of cells. So the cells that grow in full light tend to be smaller and less undulated than cells grown in the shade. And in fact, there's even an equation for this, the so-called undulation index, set up by this paleobotanist, Wolfram Kirchner, to measure the degree of undulation and correlate that with the whether it was a sun leaf or a shade leaf. But what we wanted to do was to actually use phytoliths to reconstruct light regime. And we can do that because epidermal phytoliths are often a direct cast. It's just a copy. It's an infilling of an epidermal cell. So the shape of that epidermal cell will be reflected in the phytolith and the size. And we wanted to look at phytoliths that we extracted from a soil sample or a sediment sample and look at the distribution of those shapes and sizes and use the average to say what kind of light regime was present at this site. So, and I should say that what we wanted to reconstruct is a measurement called the leaf area index, which is basically a measurement of openness. It looks at the area of leaves relative to the area of the ground. This is a hemispherical photograph taken straight up. And you can see here in this habitat, we have very little leaf tissue covering the sky. Whereas in this forest here, we have a lot of leaf area covering the sky. So this is a measurement of the general openness of a habitat, of the light regime. So to calibrate this method, Reagan went to Costa Rica and she looked at soil phytoliths from different vegetation types there, rainforest and a dry forest. And she also took hemispherical photos so she could measure leaf area index from these photos. And then she saw how those things correlated. Specifically, she saw how the actual leaf area index correlated with leaf area index predicted from the shape and size of those soil phytoliths. And she found a pretty good relationship there. And so we used that relationship to reconstruct leaf area index in fossil assemblages from southern South America. So the same ones I've been talking about. So here we have time on the x-axis moving forward to the right, and we have reconstructed leaf area index on the y-axis. And here are some examples of typical modern ecosystems that have particular LAI ranges. And what we see here is that as we go from the early to middle to late Eocene, we see a drop in average LAI. So an opening of landscapes down to values that we now associated with desert and shrubland. So very open habitats. And for the most part, things remained fairly closed until sometime in the middle Miocene, when we see a regreening, a spread of forest there. So if we compare the floras that I showed you earlier with the measurement of openness, we see that when we reconstructed it, we see that we go from closed forest to a open shrubland. And because we have a lot of palms here, we reconstructed this as some kind of non-analog palm-dominated shrubland, something we don't have on Earth today, because palms tend to grow in wetter climates. And then it's not until here, like I said, in the middle Miocene, that we start seeing some closed forest and then again an opening. And during this whole time, like I said before, we tend to generally have very little grass. So Patagonian were covered in this open vegetation, low-end grasses for most of the Eocene through the Miocene. And if we correlate this with the evolution of Hypsodonty and Hypsilodonty, we see that there's a broad correspondence between when we see opening of the landscape and when we start seeing these Hypsodont taxa. We also had some geochemical data from stable carbon and oxygen isotopes from our geochemist here, Matt Cohn. And basically, to make a long story short, this is such a complicated graph, don't worry about it. We found that we had stable, warm, and arid climates starting in the early to mid Eocene until the early Miocene, when we start seeing wetter climates. So we hypothesized then that the opening up of vegetation that we see into these shrublands in Patagonia might be due to this aridification during the mid to late Eocene. So our alternative hypothesis then for Hypsodonty is that it evolved in open, dry habitats in response to not feeding on grasses, because there were not that many, but instead ingestion of grit linked to either intensified soil erosion or influx of ash. And as I mentioned before, the sediments that we're looking at have a lot of volcanic ash. So we know that there was a lot of volcanic ash in the environment during this time. Okay, so to conclusions from this part here is that this evidence from South America is mirroring what we see in North America, namely that open habitat grasses evolved and diversified long before they became ecologically dominant, at least in these two different regions. And also that Hypsodonty evolution is not linked to grasslands, but more likely to just generally speaking, open, arid and dusty vegetation. And I want to say that this sort of simple idea of how grasslands are spreading in response to climate is not true, but instead we see that the timing and nature of grassland ecosystems, ecosystem evolution varies depending on the continent and area. So we really need to look more closely to fully understand the drivers here. Okay, so in conclusion, I want to just state here that grassland ecosystems on different continents have unique and complex histories. And this is something that's important to consider when we think about basically conservation and how we manage ecosystems moving forward. There's been a big push to basically plant trees everywhere, and implicit in that is viewing grasslands as a basically disturbed forest, so not as unique ecosystems with a long evolutionary history. But the work I just presented negates that. It rejects that idea that grasslands is a very recent thing, but in fact shows that they are ecosystems with a long history, and that history is something that's important because it has led to the evolution of unique faunas and interactions with climate and all that that I talked about previously. And so we need to continue, again, study these ecosystem histories across continents to understand better how they came about, why they came about, and how those factors shape their current function and future responses to climate change. So we're continuing to do that. And I just want to highlight some ongoing work on grassland evolution. Well, just to say that we are continuing to work in different parts of the world to expand and refine the record of grassland evolution. In the next few weeks, hopefully, we'll publish some work from Eastern Africa that I didn't have a chance to talk about. But yes, we need to continue this work to refine and expand the record of grassland evolution. Another thing that we're doing is trying to refine fightlets as a paleoecological tool. So right now we can use fightlets to talk about broad groups of grasses within the grass family, but we need to get more specific to be able to better understand, better reconstruct how grasses have diversified and the ecology of past grasslands. So to do that, my former postdoc, now collaborator, Tim Gallagher at the Bishop Museum in Hawaii, and a whole slew of undergrads and some grad students have been working to study the evolution of phytolith three-dimensional morphology within grasses. And we're really hoping to come up with a detailed key, if you will, for fossil phytoliths across Poesi. So just to highlight some of that work, we have quantified three-dimensional shape, like I said, of grass phytoliths from modern grasses across Poesi. And to do that, we use confocal microscopy, which gives you basically pictures slicing through a phytolith. And then we can use different programs to stitch that together to a 3D model. We can then basically measure that morphology using something called 3D geometric morphometrics and machine learning to analyze the shape and size differences between different grasses and link that to the group within grasses that they belong to, so to phylogeny. And this is just showing some examples of grass phytolith in situ, so in the grass epidermis, and this is what the 3D models look like. And so we've been able to do this. This is showing a paper that we published a couple of years ago, looking at some of the early divergent grasses and forest grasses, so bamboos and rices and things like that. And this is showing these 3D shapes, how they map onto a circular phylogeny, family tree. And these pie charts here are showing the likelihood of a certain morphology at these ancestral nodes. And so what we found is that phytolith shape and size varies with phylogeny in the Poesi. And also we're able to reconstruct the ancestral grass phytolith shape as one of these elongated so-called saddles. We've also, and this is kind of the primary purpose of this, is to try to identify and place taxonomically fossil grass phytoliths. So as a test case, we took some of these Eocene phytoliths from Nebraska that I had previously determined or hypothesized were from bamboos. And we used our modern data set combined with the fossil data set, and we subjected them to different statistical or ordination methods and machine learning, something called linear discriminant analysis. Basically, it is trying to use statistics to figure out which group of grasses these fossil phytoliths belong to. And what we found was that these phytoliths were indeed most likely woody and herbaceous bamboos that we have there in the Eocene of Nebraska. So basically confirming what we previously thought, but also refining what we previously thought. So showing that there was some diversity in the phytoliths that we found back then. And now we're continuing to look at the rest of the grass family. We have over 4,000 3D grass phytolith models from almost 200 species from across Poesi. And we're analyzing data from Poeti right now, and also working with a whole bunch of undergrads again to assemble all the rest of the data for the PacMad clade. So stay tuned. And with that, I just want to say thank you so much for your attention, and I'm happy to take questions.
Thank you for that wonderful talk, Dr. Strömberg. And we do have a lot of questions, but I'm going to use my moderator privilege to ask you the first question, and it's going to be a simple one. So a lot of early paleo art included grasses with dinosaurs, and for a long time afterwards, when we understood the fossil history a bit better, it was determined that the grasses were in error. But with the evidence of the titanosaurs eating grasses, would you suggest that grasses should be with moderation, reintroduced into Cretaceous paleoart?
Not, I would say not unless it has been established in a particular place that they were, locally abundant or even, more than regionally abundant. I don't think there's any evidence that they were regionally abundant anywhere during that time. I said that grass fossils in general are quite rare, and that is true. But we do have what we think are grass pollen, throughout the Cenozoic, and in the Miocene, in the more recent times, they are very abundant in some sediments. So I think if there were abundant grasses in the environment, we would have seen them in the pollen record. There would have been some indication. It's possible some of the more recent work we've done in India seems to indicate that they may have been more abundant right at the beginning of the Paleocene locally, but I would not assume that they were abundant just because they had evolved that time.
Great, thanks. All right, so our first question from our attendees: has the transition from C3 to C4 and CAM pathways resulted in the widespread advent of grasslands? And if yes, can the increased productivity of the C4 pathway and a lack of the C2 pathway contributed to this?
Great question. So I didn't talk at all. I meant to not even mention the different photosynthetic pathways, but I forgot to get rid of the C3 there. But it's a great question. So within, for those of you who don't know, within the Pac-Man clade, these tropical grasses, we see the advent of this C4 photosynthesis maybe as many as 30 times independently. And C4 photosynthesis is very advantageous in hot and dry climates with summer rain in particular, and at low CO2 levels. And so it's often been thought that, C4, okay, so there's that. Then also in the fossil record, using geochemistry, using stable carbon isotopes, we can see when C4 grasses become abundant, because C4 grasses have a different isotopic signature compared to C3 vegetation. So C3 is the ancestral photosynthetic pathway that most plants have. So people often assumed that the evolution of grasslands was synonymous with the spread of C4 grasses. But now we know, based on fightlets, and I should also say implied also by Hypsidanti, but definitely shown based on fightlets, that prior to C4 grasslands in many regions, we have C3 grasslands or grassy biomes, grassy vegetation. And also, so for example, in North America, what I showed you was the evolution of grasslands there, or grassy biomes, by say 22 million years at the latest. And then it's not until 5 to 6 million years ago that we see the spread of C4 grasses in North America. So there's a lag there, where you first had C3 grass dominated habitats and then some C4 grass dominated habitats. In other continents, there's less of a lag, but there's often some indication, pollen or fightlets, that there was C3 grassy habitats appearing first. Not everywhere necessarily, but in some regions. So they're not necessarily synonymous.
Great, thanks.
That's all of that? Yeah, okay.
Yeah, I think that covered it. Is there a functional reason for the difference in phytolith shapes between different plant groups or families?
Well, a great question. If we think outside of grasses, oftentimes those phytoliths are a direct reflection of the types of cells, because they're basically infillings of those cells. And that's why they're less diagnostic outside of grasses. And also that kind of, the function is just basically tied to the function of the cell. So you can have, in the epidermis, you can have stomata, so the pores, they can be silicified. You can have the vascular tissue being silicified. You can have regular epidermal cells. You can have hairs being silicified. So those are just, there's no function outside of just what the function of the cell. Within grasses, we see shapes that don't seem directly tied to the shape of the cell. They're sort of independent of the shape of the cell. And that's where people have talked about potentially different functions relating to the shape. So there's some that are very elongated, some that are short or bilobate shape and things like that. There's no great sort of strong hypothesis, I would say, at this point about the shape. The most interesting, compelling thing I heard was when I talked to a structural geologist who suggested that the fightlets that are long and sort of have sinuous margins or wavy margins, that maybe they are similar to how minerals behave in metamorphic rocks when there's a lot of tension. They tend to be sort of, I forget, boudinage or something like that, what it's called. And so he suggested that maybe they have that shape to strengthen the grass leaf when there's a lot of wind or other sort of forces moving the leaves. And that is fascinating suggestion that has not been tested. But other than that, we don't know. So for most plants outside of grasses, the fightlets are simply when silica-rich water is moving through the plant and evaporating in the leaf surface, the silica is deposited in those cells. And so, no direct function of those fightlets.
Great. So there's a follow-up question to that one, and you've already answered it partially. So the follow-up question was, are the phytoliths purely an anti-herbivore measure, or do they serve other purposes? So you've already touched on that. But the sort of extension of that is, does their abundance in plant tissues or their shape tell you anything about the predation stresses that the plants are being subjected to by local herbivores?
Also a great question, and something that we've done some work on. We did some work where we tried to map onto a phylogeny the relative abundance or the concentration of silica in tissues, both across land plants as a whole and then focusing in on grasses. Indeed, silica has been proposed as an anti-herbivore defense. And so you would expect to see either silica being increasingly deposited within a group of plants that's co-evolving with herbivores, say, so grasses would be an example of that, or during certain time periods, like say when we see the advent of these tooth batteries in some dinosaurs that they maybe were feeding on abrasive things. So far we have not been able to see any patterns of silica deposition in plants that support those hypotheses. For if we look broadly on plants, I think we just don't have enough data and enough precision in when silica evolved. So silica deposition evolved, probably was present or possible with early land plants onward, but we'd see it sort of becoming more abundantly used within different plant groups at different time periods. So it's not that all plants are just doing it a lot or a little, but there's sort of certain groups within, say, flowering plants or within conifers or whatever that are depositing more silica. But so far, we don't see any great patterns there. If we look within grasses, we also don't see patterns of increased silica deposition, for example, in open habitat grasses, which we think were interacting with herbivores in a more sort of meaningful way than a forest grass. Forest grasses are just not abundant enough to sustain a lot of herbivores. So it's not, we wouldn't expect to see that as much, but we don't see those patterns. We see instead that silica deposition seems to be more linked to the climate. So in climate with a lot of evapotranspiration, so a lot of water is being moved through the plant because there's a lot, it's dry and a lot of evaporation. That's where we see more higher levels of silica. So it seems more, not necessarily that there was selection for that, but maybe it's something that's happening, in the plant, in each individual plant, that they're basically sort of passively taking up a lot of water, a lot of silica, because a lot of water is moving through the plant body. Sorry, that was maybe a little bit confusing. The short answer is, we still think that fightlets are an anti-herbivory defense, but we don't think that it was an anti-herbivory defense to large ungulates, more likely to insects. And that is a lot harder to test because we don't have a good fossil record of insects. So, that's something that needs to be looked at.
Great. Okay, so the next question. Have you or has anyone carried out a study of phytoliths to understand herbivore diversity in regions like China or Russia?
Well, Russia, I don't think so. Certainly we haven't, and I don't think there's anyone working on the timescales, sort of like, deep time the way we are. In China, I have actually tried to find fightlets in many sediments and have been unsuccessful, except in one place. But it was just one lake. And so can't really address that question with just one place. And I think I reviewed a paper once that was looking in, I think it was Mongolia, Inner Mongolia, or Tibet, one of the three, where they had a record of fightlets. But I don't know whether it got published. Anyway, so the short answer is that there is work ongoing looking at that. But I don't think that paper directly compared it to herbivores.
Okay, great. Do you see any relationships between phytoliths in the fossil record and the incidence of wildfires?
This is a question I'm hoping to answer in collaboration with folks at Alison Karpen, Kate Freeman, who are our geochemists working on different ways to look at fire in the fossil record. And the short answer is that we don't have a record where we have both fightlets and those kinds of fire indicators. That said, we are making them. For example, I'm working in Australia, and there we can make those comparisons. We just haven't even really began the fightlets counting yet for that. There is also, because fightlets are affected by fire, so they get black and slightly deformed. So we have used it in Patagonia to look at fire across the Eocene-Oligocene boundary, where we saw that there was a change in the fire regime, or we inferred that there was a change in the fire regime, looking at fightlets and some other geochemical proxies. So we don't know yet. We just haven't reconstructed the record from Australia, for example, to say what the relationship is there. However, I will say that the record from Australia looking at pollen and these fire indicators indicate that the fire regime there, which is very strong, was established before grasses and long before C4 grasses. So there's probably not a strong correlation, but in other regions, I expect there are. For example, in India and Pakistan, I think there's a reason to think that. But yeah,
great. Do you have time for a few more?
Yeah, sure. Yeah, sorry that I'm so long-winded.
No. Can you comment on the biogeography and the distribution of extinct members of Poaceae in the Cenozoic? And can that information at all be used for the future of agricultural practices?
So the short answer is that I think generally speaking, we will be able to say something, like I was talking about, understanding a little bit about sort of the ecologies of different taxa, how the distribution of different taxa with certain ecologies have changed through time, can maybe tell us about the breadth of those ecologies. So right now we have grasses, certain grass groups living in different habitats and in different climate zones and whatnot. And so we then assume that is their climate regime that they are most adapted to, and that's the kind of habitat they are most adapted to. But in the fossil record, if we find these lineages or these groups of grasses in different climates and habitat types, that might tell us that their fundamental niche, if you will, like, so where they can live, is actually much broader than what we see today. Then that is helpful, I think, for being able to analyze what's going to happen with crops, for example. Of course, crops are very altered from their ancestors, in like genome duplication and all kinds of selection happening. But the fundamental things, so like C4 photosynthesis in corn, for example, we don't know exactly why C4 grasses became exploded onto the scene very late in the Miocene, as opposed to earlier in the Miocene, or even Oligocene or Eocene, when C4 photosynthesis evolved. And so we're trying to figure out what were the factors that made it advantageous to be a C4 plant. If we can determine those factors, we know more about what to expect in terms of where C4 grasses can grow productively. And so that will be directly informative for agriculture, I think.
Great. We'll do one more and then we'll let you go. Do you have any ideas or have thought about creating a theoretical morphoscape using phytoliths based on mathematical frameworks similar to those done on foraminifera?
I think the answer is yes, in a sense. That's a little bit what we're trying to do with this 3D work that I mentioned. I don't know exactly what sort of what the morphoscape for forams is. I don't know, like, is it sort of looking at evolutionary adaptive peaks or something like that, or sort of just looking at how forams have evolved in this morpho space or something like that? That's certainly kind of what we're trying to do with the 3D work I mentioned, but I don't know exactly how comparable it is. I think the holy grail we're trying to go for is to map the 3D shapes on a phylogeny, but that is not computationally possible, or at least it hasn't been in most recent years. So it's something that we need to work with a computer scientist or something to try to figure out how to do that. Yeah.
Great. Well, thank you very much, Dr. Strömberg, for your time today. We really appreciate it. We hope you have a nice day.
Thank you. My pleasure.
You're welcome.
Have a nice day.
All right. Bye-bye.
Bye.